Hello everybody, Mr. P here. Chapter 8, Lecture 4 already. And in this lecture we're going to talk about the light-dependent reactions, which is the first half of photosynthesis. And in the previous lectures we've talked about the foundation, like chemiosmosis and substrate-level phosphorylation and redox reactions and light absorbance and reflectance. And we've talked a lot about electrons being energized and electrons giving off energy and the electron transport chain. And so we're going to put all of that stuff together, and we're going to try by the end of this to understand the light dependent reactions in a lot of detail. It's really going to require you to buy in and take good notes, listen carefully, and ask good questions in class if there's something you don't understand. This is a difficult process, but it's one that absolutely must be known and is something that you're going to be tested on in about a week and a half. So, light-dependent reactions require sunlight, and that sunlight, like I've talked about in previous lectures, comes to the Earth in photons. Photons are particles that travel along a specific wavelength that is emitted or are emitted from the sun and will travel great distances to get to the Earth. But these photons are going to be collected by chlorophyll pigments that are embedded in a membrane, specifically embedded in Photosystem I and II, that are located in the thylakoid membrane of every chloroplast, and it's these photons that basically give rise to all glucose on the Earth in the form of photosynthesis. Photosynthesis converts photons into glucose molecules that provide photoautotrophs with not only their glucose, but provides all of the consumers with the glucose they need by consuming those autotrophs. Okay, little meme for you. A photon checks into a hotel and is asked if he needs any help with his luggage. And he says, "No, I'm traveling light." Get it? Because he's traveling a wavelength from the sun, which is actually light beam. Photosystems. Photosystem I and II are both going to have a substantial amount of chlorophyll, and we're not necessarily talking about the actual photosystems at this point, but these chlorophyll molecules can be looked at as kind of like a little solar panel. And the job of chlorophyll and any pigment really is to absorb sunlight. And so these photons, which are going to be traveling along a wavelength, are going to be absorbed by these little solar panels, and the electrons that are sitting here at this molecule, say 2 electrons, because they typically will travel in pairs, these electrons are going to sit on this little kind of docking station called the chlorophyll molecule until they're energized enough to be used to produce other compounds. And so these electrons are going to sit at these chlorophyll molecules and are going to be energized over time. And it doesn't take that long, but these electrons are going to be energized by the photons that are absorbed by chlorophyll until they gradually increase in energy to a point where they eventually can do work and produce other compounds. So photosystems I and II are going to capture light energy in the form of photons. They're going to energize electrons, or accelerate, lift, however you want to say it, electrons to a high energy level. And once these electrons are at a absolute highest energy level possible, then they have a little bit of energy to give off, and it's this energy that they give off that will allow us to do work. Photosystem II, like I've said in class, is actually the first photosystem. So Photosystem II is the first photosystem, okay, and that's going to be key. First photosystem is Photosystem II. Second photosystem is Photosystem I. Okay, this picture shows the entire process. It starts here at the beginning, and it travels through Photosystem II first, into Photosystem I, and then ultimately to our final electron acceptor, which I'm just going to say FEA. Final electron acceptor, and we'll go through this whole process. But water is going to start the entire process, okay? And so we start with water, and we always are going to start with a hydrolysis reaction. And hydrolysis takes water, which is H2O, and it's going to split this water molecule using the light energy that is bouncing around between all of these pigment molecules until it hits the water that's sitting at reaction center P680. Once the light energy, which is very intense, hits a water molecule, which is a tiny little molecule, it's going to blow that molecule up into three things. And the three things are 2 hydrogen ions, 1/2 of an O2 molecule, which just means 1 oxygen, and 2 electrons. Okay, so you're going to split H2O up into three things: 2 hydrogen ions, 2 electrons, and 1/2 of an O2 molecule. The O2 molecule is going to be given off to the atmosphere. Okay, that's why plants basically release oxygen as a byproduct. Where did the oxygen come from? The oxygen came from water. Every bit of oxygen that we breathe that was produced by a plant is going to come from water. That oxygen used to be locked in water. The plant unlocked it from water using the sunlight as an energy source. These hydrogen ions, we're going to use them later. They're hanging out. Those are going to be really important. The electrons, however, is what we're going to start looking at. P680 is the reaction center, or the primary electron acceptor, within this photosystem II protein complex. All of these lighter green colors would all be chlorophyll B pigments, and this darker green one, which is the primary electron acceptor, would be chlorophyll A. Okay, so I know in class we've talked about chlorophyll A and chlorophyll B. Chlorophyll A's are kept within the reaction center of each photosystem. Chlorophyll B is kept within the outside of those particular photosystems. So chlorophyll A is kept right here, chlorophyll B is kept everywhere around it, and together chlorophyll A and B both will absorb the blue violet, and the orange red spectrums. Remember, chlorophyll A is going to be the red and violet spectrums, and the B, or chlorophyll B, will be the blue and the orange spectrums. Together they pretty much absorb everything but green and yellow, which is why plants appear green and yellow. And that's because they're reflecting green and yellow light. Once the electrons are split from water and sit at this reaction center P680, all of this light energy is going to be funneled to this reaction center, kind of like laser beams bouncing off a mirror. If you have ever seen any kind of video or movie where they're taking laser beams and using all these mirrors to kind of like bounce the laser beam around a room, that's kind of how the photosystems work. All of these photosystems are going to be structured so that they funnel this light energy right to the reaction center. And when the electrons are sitting here at this reaction center, all this light's going to be energizing them. And we kind of represent that by this arrow. See how the arrow goes from very skinny arrow to a much wider arrow? And that's to represent these electrons absorbing energy and becoming very much energized. Once these electrons are completely energized, then the electrons will enter the electron transport chain. And remember how they do that. Those electrons are going to reduce this first protein. Remember, these are proteins. And reduction means gaining electrons. So they're going to reduce this first protein. And as they do that, they're going to release a little bit of energy. And then they're going to oxidize that protein and reduce this protein. And at that protein, they're going to release a little more energy. And then they're going to oxidize the protein and reduce this protein. And release a little bit of energy, and then oxidize the protein, reduce this protein, release a little bit of energy, and then oxidize the protein before being shuttled to this second reaction center. Now this reaction center is the reaction center of Photosystem I, and that specific reaction center is P700. The reaction center here, remember, was P680. So reaction center of Photosystem II is P680. Reaction center of Photosystem I is P700. Again, all of this light is going to bounce around all of these chlorophyll B molecules until they reach the electrons, which are sitting here at reaction center P680, or P700, excuse me. And once the light energy hits those electrons, it's going to energize those electrons again. Remember, kind of think about these electron acceptor locations as kind of a battery recharging station, and electrons are kind of like batteries. We're going to energize this battery initially. It's going to move to a high energy state. We're going to do a little bit of work with that electron, gradually release the energy that's locked in that battery for work, and then we're going to charge them again. And once we charge them again, we're going to send them to the second smaller electronic transport chain. And then our final electron acceptor, which is NADP+, is going to pick up these two electrons with a hydrogen ion and be converted to NADPH. And we've talked about that molecule numerous times. NADPH will then travel to the Calvin cycle. And I'm just going to put CC for Calvin cycle. That is the dark reactions. Now, what is this energy that was given off by those electrons used for? It's going to be used to actively pump H+ ions. If you think about chemiosmosis that we learned, this is the energy that is going to be used to actively pump those H+ ions from the bottom of the membrane, which means the inner cristae, the cristae membrane, the inner matrix of that mitochondria. But it's also going to move the H+ ions from the stroma of the chloroplast into the inner mitochondria, or the inner membrane space of the chloroplast, okay, inside of the thylakoid. So photosystem 1, just as a repeat, has P700. Photosystem 2, remember, had P680. Those are the primary electron acceptors. Those are specifically chlorophyll A, and all these little lighter ones around the outside would be chlorophyll B. The final electron acceptor we talked about was NADP+, that is nicotinamide adenine dinucleotide phosphate. And when you put that together with an H+ ion and electrons, you fill the taxi and you produce NADPH. And where does NADPH go? It's going to go to the Calvin cycle. Okay. So, hydrolysis reaction, this is the reaction we started with. The hydrolysis reaction is the very beginning of the light-dependent reactions. And again, it is one of the light-dependent reactions because what is the energy source that breaks the water down in the first place? It's light energy. So water is brought into the chloroplast through osmosis. That is water moving against or through a membrane from high to low concentration. And that water is going to move into photosystem 2, specifically reaction center P680, and is going to meet a big intense beam of light that is funneled through all the thylakoid membranes and all the chlorophyll molecules, and specifically chlorophyll B molecules, to that P680 location, where it's going to split the water molecule into 2 H+ ions, an oxygen, and 2 electrons. The electrons are going to go to the electron transport chain, oxygen is going to be released to the atmosphere, and hydrogen ions are going to be sent through our ATP synthase enzyme to produce ATP. Okay. H+ ions are sent inside the thylakoid membrane. That is our high H+ ion concentration gradient. And like I said, oxygen is going to go to the atmosphere, and electrons will be used to do work. Here is the entire light-dependent reaction process, and this includes our ATP synthase enzyme, which you should know about at this point. This includes the chemiosmosis portion of it. I'm going to go ahead and go to the next slide, which has a bigger picture, and we'll go ahead and work through the process again. This big purple circle would be photosystem 2, and I can label that PS2. And the next big purple circle would be photosystem 1. And this is our hydrolysis reaction. Hydrolysis is when we take H2O using light energy and we split it into 2 electrons, 2 hydrogen ions, and an oxygen molecule. The oxygen is going to be released through the stomata of a plant and be released to the atmosphere. So I'll just put ATM, atmosphere. H+ ions are going to stay out here in this thylakoid compartment, which is our high H+ ion concentration. Our low H+ ion concentration is down there in the stroma, and our light ultimately is going to hit our chlorophyll molecules and be shuttled to our electron acceptor location, which is P680, The electrons are going to sit at P680 until they are energized to a high energy state, at which point they will enter our electron transport chain. And as they enter the electron transport chain, they're going to walk down the series of proteins that are gradually releasing energy, and that energy, like I said, is going to do work. All of that energy is going to be used to actively transport H+ ions from the low side to the high side. And remember last chapter when we talked about active transport is moving solutes or moving materials from an area of low to an area of high concentration against a concentration gradient using energy. And this energy right here would be, just write energy. Okay, energy. That energy is going to be given off by the electrons gradually as they move down the electron transport chain. As soon as the electrons enter P700, which is the electron acceptor at photosystem I, they're going to be hit with a little bit more light energy because remember they are the batteries that were just depleted. We have to recharge them again, and so they sit at P700. This light molecule or the light photons are going to bounce around until they hit P700, energizing the electrons to a high energy state. Those electrons are then going to bounce down the second smaller electronic transport chain and meet NADP plus H plus, and together with the two electrons are going to produce NADPH. NADPH is going to travel to the Calvin cycle where the glucose will be made. So that is the I guess flow of electrons. They go from water using light, producing energy that is going to move and maintain our H+ ion concentration gradient, eventually accepted by NADP plus to convert it into NADPH, which will take it to the Calvin cycle. The Calvin cycle happens down here in the stroma, so it's just going to kind of go further into the stroma where it'll meet that cycle and eventually produce glucose. Now, we have a lot of H+ ions up here. Okay, this is our high side, and so H+ ions are going to flow down through our ATP synthase. Just like in chemiosmosis, they're going to phosphorylate this ADP molecule by adding a phosphate. And so when these substrates go into our enzyme, they're going to come out as ATP. This too will go to the Calvin cycle. Okay, because the energy we produce in the form of ATP and NADPH are going to be used in the Calvin cycle. This is the ATP synthase portion. This is photosystem I portion. This is photosystem II portion. This is the electron transport chain portion. This is the hydrolysis portion. So, hydrolysis, photosystem II, electron transport chain, photosystem I, ATP synthase, and the ATP production portion. Okay. If you have questions about this process, this is not the last time you'll see it in class, but if you have questions, make sure you bring those questions. We're going to do a lot of drawing. We're going to do a lot of describing. We're going to do a lot of writing. Make sure you understand this process by the time the test comes, because every part of this process will be assessed on in some form or the other. Okay, and so I think that's where we'll leave it. In order to leave you with a joke, I thought I'd throw in photosynthesis. Mr. P, out.